welcome everybody to this is number 5 in the series of webinars from Project 81, Work Package 7, Developing Surge Capacity. We've covered a number of topics in the past. Today, we're going to look at pathogen sequencing, detection and sequencing, and we've got a full agenda in front of us, which you can see. So let me introduce myself first. I think many of you will know me. I'm John Jones. I am the team leader, key expert 1 from SCGS Europe, leading Project 81, which is improving biosecurity in Southeast Asia. Been running for a couple of years, but of course the pandemic has slowed us down, so we haven't been able to do any face-to-face activity. So we've been running these webinars on a monthly basis, and I hope we may be able to continue this into next year, but I'll talk about that later. So we're going to have some welcoming remarks from Attorney Manalastas, and then I've got 3 presentations for you, and I'll introduce the speakers as we go along. But we've got an introduction to genome sequencing, then we've got a session on metagenomics, so a bit more detailed from Christopher Logue, and then a final one from Joris in the Netherlands, from RIVM, about wastewater. But we've got a full package. So before I do anything else, obviously your microphones are muted. If at the end you want to ask questions, it is possible for me to put you on the video if you want to be, to ask your question verbally, or to use the chat. I'm going to launch some polls during the session, some question and answer things, just to make it a bit more interactive and get a good idea of your interests and what you want to focus on, and you know, your level of understanding of some of the material. So it's helpful for the speakers to know who we've got in the audience. So I'll launch that in a moment. But first of all, I'm delighted that we've got Attorney Manalastas in Manila, who's going to offer the opening remarks for us from the head of the Regional Secretariat. Always look forward to hearing what Attorney Manalastas has to say. So welcome to you, sir. I'll hand the floor to you if you'd like to take over, and I'll stop sharing the screen. And yeah, over to Manila. Thank you.
Well, good morning, colleagues in Europe, and good afternoon, colleagues in Asia. I am Attorney Florentino Manalastas Jr., the alternate head of Secretariat in the Southeast Asia. Of course, there are 3 of us working on alternately, depending on our availability, but we always coordinate. Of course, the primary is Undersecretary Picoy Cascolan, our Executive Director of the Anti-Terrorism Council Program Management Center. And the second alternate head, whom you see very often, is Director Rolando Aquino. So there are 3 of us working on the same position. I would like to greet Mr. John Jones, the team leader of Project 81, and of course, Mr. Alexander Hamilton, Uniquely CBRN Regional Coordinator, whom I had the pleasure of finally meeting here in Manila. The national focal points, partners, guests, and participants, thank you for joining today's webinar. We are here gathered again for another webinar organized under Project 81, which aims to strengthen biosecurity measures in Southeast Asia through multi-sectoral involvement. Today, we shall hear from subject matter experts who will discuss novel pathogen detection and sequencing. It is intended for public health practitioners, laboratory managers, and colleagues from Health Ministry. Lessons from the past outbreaks and pandemics tell us that prevention is always better than cure. These outbreak control measure and disease prevention efforts rely on swift identification and characterization of new and existing pathogens. Once we know what we are dealing with, we could respond adequately. This is the basic philosophy being applied by doctors in treating patients. In the case of the novel coronavirus or SARS-CoV-2, the viral agent responsible for the present pandemic we face now, it took us some time to finally know its characteristics, including how it spreads, and therefore it has created havoc globally, claiming the lives of millions of people. But the good thing is, through international cooperation and efforts in open research, we have made significant progress in fighting the virus, which includes quick development and mass rollout of vaccines, among others. I would like to stress that the rapid developments in science and technology should support sustainable development and should not be used for the proliferation of weapons of mass destruction. It is in this context that the work of the CBRN Center of Excellence becomes truly important. We try to shed light on successful strategies and activities to mitigate CBRN risk, while further advancing and strengthening international cooperation programs and initiatives in this regard. With that, we hope this seminar will cater to your needs and provide you with relevant information to mitigate biological risks and threats. On behalf of the EU CBRN Center of Excellence Regional Secretariat for Southeast Asia, I welcome you to today's webinar, and I hope you will find the discussions insightful. Thank you very much, and I turn over the floor to Mr. Jobs. Magandang araw sa inyong lahat. Mabuhay.
Thank you very much, Attorney Manal. Oh, am I in? No, I am unmuted. Thank you very much, Attorney Manalas. That's excellent. Always good to hear from you. And yes, we're going to look at some new developments in science used for very peaceful purposes and some important stuff that's coming. So yes, thank you for underlining the importance of the work that we're doing in the Centers of Excellence and in particular in Project 81. So thank you so much, sir, for that. So what I'm going to do now...
Just had change, full stop.
Oh,
in fact, we will need to,
Stuart,
in reality, put together
Stuart,
a new
I'm going to mute
SOP regarding payments for consultants.
There we go. Thank you, Stuart. So let me launch some polls. What I'd like to do now is just to introduce you to the polling system. Some of you are familiar with it. Some quick questions and answers to find out a little bit about yourselves, your role and where you operate and so on. And well, it'll be obvious once I launch them. So I'm going to put the poll up now. This first one's coming up now. Here we go. Hopefully that's now visible. So a few questions there. Lab technician, are you a lab manager, a policy official, clinician, researcher, or other? And I'll give you a couple of minutes to get that one going, and I'll share the results. Coming slowly, we've got about halfway through, 60% have answered at the moment. Just wait for a few more. I normally give this a couple of minutes just to allow people to have a chance to answer. If it's a difficult question, I'll give you a bit longer, but this one should be straightforward. Okay. A few more coming in. We're about 80% now. Still a few more answers coming in. We've got, I'll share the poll results in a moment. We've got quite a lot who have put themselves down as other, so that's interesting. That means I haven't quite got my questions right. Maybe I should put some extra categories in there. 83%, 54. Okay. Okay, I'm gonna end the poll now, and I will share it with you. And so there we are. We've got a good mix. Got 20% roughly lab technicians, lab managers, policy officials, clinicians, researchers, and others. So thank you for that one. So that gives a picture of who you are. Now the next one is going to be your experience of previous outbreaks. And here it comes. So, which of the previous outbreaks have you experienced? I've deliberately taken SARS-CoV-2 out of this. So SARS-CoV-1 is there, not SARS-CoV-2. We've got MERS, H1N1, Zika, and anything else that you care to mention. So, morning, Alex. Hi, Alex. How are you?
Very well, John. Yes, it's 4:45 p.m. here.
Okay.
But,
Yeah, we're running a bit late, aren't we? This is because of the hour change in Europe. Unfortunately, we need to stop doing that kind of stuff, I think.
Oh no, we really appreciate, in fact, you accommodating Southeast Asia schedule. I think it's very, very much appreciated. I know it's still early there in Europe.
That's okay. I'm going to put you on the spot now, Alex. Which do you think is going to be the #1 answer out of that one? I don't know how you voted, but what's your guess?
In terms of the most impactful disease event, is that?
Well, no, just one is which have you experienced out of those? I mean, maybe none.
Well, I mean, certainly SARS was a very impactful event, not only in parts of Southeast Asia and Asia itself, but also in Canada. I was in Toronto, a large cluster of cases there, and it certainly was very impactful. And I also recall very well the swine flu pandemic. And of course, I think nothing has been more impactful and in all of our lives than COVID-19. So. Oh,
that's true. That's very true. Okay, yeah, you've done quite well at covering all of those bases there, Alex. So I'm gonna share the results now, and you're close. Not completely right, but you're close. Here we go. Right, there you go. There's the results from the audience. So a lot with H1N1. That's not a surprise, I guess, because of the region, but a good spread across, including Zika and a few others there which we haven't listed out. So thank you for providing that with you, and apologies, Alex, for putting you on the spot, but there you go. Let's see the results. Yeah, good spread across all of them. Right, final one before we hand over to Liz and I introduce her, but let's just, this is a bit more specific now, this poll. This is more to do with the topic matter for today. And here we go. So I'm not going to put anybody on the spot with this one for obvious reasons, but so let's see who we've got in the audience. Any experts? Any people fully practicing or fully competent with genome sequencing? Familiar? Yeah, okay. Unfamiliar with the concept? I'm sure there'll be a few that are not that familiar with the concept. So we've got a mix. I'll give you a little while for that to come in. I don't know if did I allow the panelists to vote on this one? I can't remember actually. No, okay. So I'll save your blushes then in that case. You can be modest or immodest as you feel like it. Okay, we're a few more coming in. Yeah. Okay. Right. I'm going to end it now. I think we've got about 75% altogether in the poll. Let me share that with you. And so this is what we've got. So yeah, we've got a few experts, one, in fact, in the audience, a couple who are fully competent, and then we've got a mix, competent with some experience, familiar with the concept but not the detail, or unfamiliar with the concept altogether. So actually, it's a good target for us. So hopefully, at the end of the session, you will be much more familiar, at the very least, with the concept and some of the detail than you were when you started. I'm not going to repeat the poll afterwards, but we will be circulating a feedback form later. So any observations about that, please share with us. Always looking to improve our sessions. So, okay, thanks very much for that. So let's stop that. Did I? Yeah, there we go. That was the final result. Let's stop that now. Okay, close the poll off. Right. Okay. So I think we're ready to go. If Liz is ready to go, I'm going to just give Liz a bit of an introduction here. And welcome to Liz. It's the first time that she's been with us. So Liz received her PhD from Cambridge University before moving to Oxford in 2010 to work on the genomics of microbial pathogens. In 2018, she joined the Mahidol Oxford Tropical Medicine Research Unit as senior bioinformatician to set up Illumina and nanopore sequencing technologies in Bangkok. She regularly collaborates with colleagues in Cambodia and Laos on the genomics of tropical pathogens. She's got a particular interest in the genomics, and I hope I can say this right, of Orientia tsutsugumushi, the bacteria which we all know causes scrub typhus. But she has worked on a wide range of bacteria as well as viral sequencing during the COVID-19 pandemic. So a warm welcome to you, Liz. And if you're able to share your slides now, please go ahead.
Okay, hopefully you can see my slides, and have you got the full screen version, not the presenter video?
Yeah, we're fine, Liz. Yeah, over to you.
All right, well, thank you for the introduction, and thank you very much for asking me to speak at the webinar today. I'm just going to give you a brief introduction to SARS-CoV-2 genome sequencing in the context of the genome sequencing we've been doing in Thailand since a few months into the pandemic. And I'll try and introduce the concepts. It's good to know that a lot of you are familiar with some concepts around whole genome sequencing, so you should be okay to follow this very quick presentation. So I think the first thing is to put this into the context of the situation in Thailand. There we go. Okay, changing slides. This is just a graph basically showing the 7-day average number of cases of SARS-CoV-2 in Thailand since the start of March 2020. And hopefully you can see my pointer, but what you can see down here is what we thought was the big first wave of COVID, which in hindsight is now a kind of a tiny first wave. Then we had a long period in Thailand where we had very few cases, and they were mostly introductions from overseas. Then we had basically a second wave of COVID starting around Christmas 2020, and that was kind of just on the wane. But then basically what happened was we had the introduction of community transmission of the Alpha variant, and then very soon after that, community transmission of the Delta variant. And you can see that's led to a large outbreak in Thailand over the past summer, which is only now kind of falling away. So I'm just going to put this, associate some of the genome sequencing we've done with this. I'll give you a little case study we did in this very first wave. Use that to explain a bit more about the concept of variants and how we decide these variants like the Alpha variant and the Delta variant and what determines those variants. And then give you an update kind of on the variants we've seen in Thailand across this whole epidemic. And what's going on at the moment. And hopefully, if I can get it to work, I'll show you kind of some phylogenies we have on our NextStrain web service. So the thing I want to say up front is quite a lot of this is genome sequencing that we've done at Moru in Bangkok, along with a bunch of collaborators. from various sites, so AFRIMS, Ramatibadi Hospital, but also there's a lot of public genome data from other sources in Thailand, from other sites that have been uploading data to GISAID, which we use to give an overview of the whole situation. So one of the key things here is that you can only put things into context through the sharing of genome data between these different centers, and also between the genome sharing that's going on internationally. So a very quick introduction. This is the genome of SARS-CoV-2. It is a roughly 30 kilobase single-stranded RNA virus, which means it's basically small enough that the sequencing method we're using is amplicon sequencing. And there are other sequencing methods, but I'm going to mainly focus on the amplicon sequencing because that's one we've been using. So this is just the kind of genome organization with the different open reading frames. And then I'm going to introduce you to this amplicon sequencing method, which is something which was developed initially in Nick Lohmann's lab, which they used to sequence Zika virus and Zika virus outbreak. They've also used it very successfully in Ebola. And all this previous work meant that when SARS-CoV-2 started up, we had these methods immediately available that could be put into practice very quickly. So it's a really simple method. Basically, it's a multiplex PCR method, but you do multiplex PCR essentially to cover the entire genome, which is possible because the viral genome is 30 kb, and so it is possible to kind of run multiplex PCR across the entire genome. So what is shown in section A here is that you essentially design multiplex primers to amplify the entire genome by tiling them across the genome so that they overlap slightly. And then what you do is you have to split those into two pools because you need to do separate PCR amplification reactions on the two pools, so that you get the full length amplicons without kind of crosstalk between the different pools. So basically you will design these amplicon primers. And for SARS-CoV-2, this same group designed a set very quickly after the start of the pandemic, which is one we use for a long time. And then for each sample, you run the two multiplex PCR pools. And then the next step is that if you have multiple samples where you have all these PCRs that have taken place, you then pool the two pools of PCR primers and add a unique index on to identify each sample. And then you pool all those samples together, and that's what you put on the sequencer. So essentially it's a mix of amplicons taken from different samples, and each sample is labeled with a unique identifier. So you can then kind of deconvolute them afterwards. So what do we do then? Basically, we put all those onto a sequencer. We've used both Illumina sequencers and the nanopore MinION sequencer for this. And you can use methods which sequence a whole amplicon or ones that fragment them into shorter pieces and then sequence them. So it's quite versatile and can really be adapted for whatever sequencing technology you have to hand. So you run them on a sequencing run. You split those reads using the unique indexes, so you've got them per sample. We then do some QC and trimming and align them to the SARS-CoV reference genome, and then call variants based on that. And this is just showing you one example of an amplicon sequencing run using these set of Arctic primers, as they're called. And so this is just showing you basically how many reads we've got for each of these different amplicons. And this is some QC we do just to check how well the whole reaction is performing. But it's known that certain regions are quite hard to amplify. And what you can see here is we have very few reads from these particular amplicons. And the dotted lines are marking kind of our minimum threshold that we would use to call variants, and our target, which is it's 50 reads and 10 reads respectively. So what it's showing is using this sequencing, we would be able to call variants across the entire genome, with the exception of a couple of amplicons where we might get dropout and be unable to read the variants in those regions. And as the pandemic has progressed, this initial set of primers have been refined multiple times to improve the problems with dropout in particular regions. There are also different primer schemes targeting different size amplicons, some with multiple sets of primers for each amplicon, basically to try and make it as easy as possible to amplify the complete genome. And the benefit of doing it by this multiplex PCR amplicon sequencing is it very effectively just amplifies the virus, and we don't get many problems with any kind of background from the rest of the human RNA in the sample. And you can also do it on the leftover samples after you've performed clinical testing. You can take that RNA and run this multiplex PCR. So what does that look like in Thailand? This is the number of sequences which have been generated and made publicly available through the pandemic. So you can see we did quite well at sequencing in March. And then there was very little sequencing. There were months where we have no sequences because there were basically no case of SARS-CoV-2. And then as the epidemic took off again this year, we've lots of sites have been trying to increase the number of sequences they've generated. And you can see we have quite a good representation for the past 6 months. Although obviously, if you compare this to the very high case levels, the proportion of cases we've managed to sequence is lower. And so that is something we need to bear in mind when we're interpreting the data, in that we're seeing a much smaller kind of proportion of the epidemic than we were in the early days. So I'm just going to introduce you to some of the analysis we can do using a paper we wrote very early on. This is from April 2020, and it uses some sequence data we generated from that first wave. So this is a phylogenetic tree of all the samples we had from Thailand at the time, and the ones in blue are the ones we generated for this paper. The ones in green are ones contributed by other groups and made publicly available. And the ones in black are ones from different countries that we use to put all the sequencing into context. And so the blue and the red here is actually designating the very two initial lineages of SARS-CoV-2. So the blue is what we call the B lineage, and the red ones are this A lineage. And what was immediately apparent is that from how these sequences are distributed on the tree, there are related clusters from different samples in Thailand. I mean, these are nearly all from Bangkok because that's where the first outbreaks were, but they're also, nested within samples from overseas. It was clear that we'd had multiple introductions of SARS-CoV-2 into Thailand very early in the epidemic. And what we also saw was we had this big cluster here where we had an awful lot of samples which all clustered together from Thailand without any other country samples. And we named this lineage A Thai, which was later renamed to A.6. And just give you a bit of detail on that lineage. At this early period, we could tell you every single mutation they had in comparison to the reference genome, because it was only kind of 10 among all our samples. And we found 3 mutations that were essentially unique to this A Thai lineage. And when this was designated as a lineage called A6, and I'll discuss how we designate lineages in a moment, these are kind of the 3 unique mutations we had in there that designated that lineage. The other interesting thing we saw even very early on is that in one of our samples, we actually had a frame shift, we had a frame shift 4 base deletion in ORF7A, which would truncate the ORF7A protein. And we found this, and at the same time, another group found a large deletion in ORF7A. And this was kind of the first indication that maybe a full length complete protein was not necessary for viral replication. And we later saw this with another sample. And so we assumed this was actually transmitted from person to person so that it could keep transmitting. So that was some very early work. I'm just going to give you a brief introduction now to what we mean by all these different lineage, variants, strains, because this nomenclature is the one that's become extremely widely used. Everyone knows what the alpha and delta lineages are now. And it was obvious from very early in the pandemic that we were going to need some way to keep track of all the different strains. So this is the initial proposal, a nomenclature proposal for SARS-CoV-2 lineages from Andrew Rambo and Oliver Pybus in the UK. And this is the kind of early tree they did. It's basically just subdividing trees. So early on, it was obvious there was a split into the A and B lineages, and then you would subdivide these based on a number of criteria. And then you would further subdivide the lineages when they basically hit the set of criteria for when you think it's useful to give them a new name. And of course, as we'll go along, we'll see that basically the A lineage is gone, most of the B lineage is gone. And in fact, it's mostly around the B.1 that is now the whole global set of SARS-CoV-2. So this is the kind of official definition. Essentially, what they wanted to do was to manage to capture patterns of virus genetic diversity and track these lineages as they move between country. But because this is an emerging situation, it had to be a very flexible nomenclature so that you could keep adding new virus diversity to it. And so that it can incorporate both new viral lineages emerging, but also viral lineages which die out and are never seen again. And this can be noted that the A lineage is now almost completely gone. And in particular, that A6 lineage we had in Thailand, I don't think was ever detected outside Thailand. So probably it shows that it was never actually detected that it was transmitted out of Thailand. Now, of course, the sequencing could be sparse. It doesn't mean it was necessarily never transmitted, but it shows that we, maybe kept control of that outbreak and didn't have it transmitted. I also quite like this paper, but it says we might expect hundreds of thousands of SARS-CoV-2 genomes by the end of the pandemic, and there's over 5 million at this point. So that estimate was a massive underestimate. So basically, how do you define a new lineage? And this is what we came up to when we were doing the new Thai lineage. Basically, there has to be a single common ancestor for all those samples. There need to be multiple samples. It can't just be in one individual. There needs to be a nucleotide change that you can designate kind of as the change you see in all those samples. And generally, there needs to be some epidemiological significance, like it shows us moving between countries, moving between, animal and human hosts, or in several cases, there's a change in transmission or virulence in this lineage. And so I'm sure that most of you know about the variants of interest and variants of concern, but just to say these are the two levels of kind of classifications of variants that WHO came up with. So the variants of interest essentially have been identified to cause transmission and with something that's predicted to affect virus characteristics. So they're kind of variants of interest are being monitored. There's currently 2 variants of interest. They get these Greek names, but I normally call them by the Pango lineages, which is the more detailed kind of A, B, and so on lineage track that I just showed you. So there were more variants of interest, but they've been removed basically as those variants are no longer seen and are assumed to be extinct. And then there's a variant of concern where we do think that there is a significant public health change, which is increase in transmissibility, increase in virulence, or decrease in effectiveness of diagnostics and vaccines. And I'm sure you've heard of most of the variants of concern, which at the moment there's the alpha variant from late 2020, the beta variant that appeared in mid 2020, and the gamma variant, and of course the delta variant, which appeared in late 2020 and then was designated variant of concern in May 2021, and obviously is the one that's fueled a lot of the recent outbreaks. So this is essentially one slide to summarize those over 7,000 genomes that have been contributed to GISAID by the people sharing public data in Thailand. And this is showing you the proportion of viral genome sequence that belong to each of these lineages over time. What I've done is highlight 2 particular lineages we saw a lot in Thailand. There was this A6A Thai lineage that we saw a lot at the early stages of the outbreak. And then our later resurgence at the end of 2020 was B13616, which is a mouthful, but it never got a shorter name. And so that was essentially the one which caused an outbreak at the end of 2020. And you can see that becomes predominant over all other lineages. And we were starting to see little bits of Delta, but these are mostly imported cases. And of course, we were specifically looking for imported cases of these worrying variants, so they may have been over sequenced. But then you see from the end of 2020, we start to get more and more cases of the alpha variant, but as the Delta variant is introduced, that then becomes predominant. And as of October 2021, it's almost all Delta variant in Thailand with just some alpha, and I think there might be a few beta cases, basically as the epidemic spreads to the whole of the country. And there's not many sequences from November, but they're all of this Delta lineage. And that mirrors pretty well, basically, this is the global situation, and you can see that what happened was that the alpha variant, starting just from the end of 2020, the alpha variant was predominant or growing, and then from the middle of 2021, it's all been the Delta variant. And this is from the Next Strain Global Build. It's a publicly accessible website that uses all the public genomes to do a kind of phylogeny and visualization. So what I'm just going to try and do now is stop sharing this screen and show you a quick visualization of all of the genomes we have in Thailand, which are Delta. So hopefully this will work. I'm doing a live demo in the middle of a webinar. But this is a Next Strain build that we maintain. which is just Delta variant viruses. And it's all the Delta variant viruses sequenced in Thailand, along with other ones that are kind of representative, basically a random selection from other countries that have Delta. And I'm just going to highlight only the ones which are Thai to give you an idea of what we've got there. So it's just going to take a sec, but basically it's going to show you, highlight only the ones which are Thailand. So only the blobs now represent only Thai strains, and everything else has kind of been faded out. And so what you can kind of see is that in the Thai Delta outbreak, although they are all Delta strains, they're actually phylogenetically quite distinct. But it looks like there have been two kind of introductions that have led to the majority of this outbreak so far. So we have this clade down here and this clade up here. And if I just zoom in like to this clade here, and I take off the country filter, you can see that these are predominantly Thai, but you can also see where they have been exported, potentially like the things that are descended from the Thai lineages are also appearing in other countries. These are in Austria. We also have a few from Cambodia that might represent transmission from Thailand through to Cambodia. And so the issue here now with all these lineage designations is that it's very useful for a public health point of view to label everything as Delta, because we know Delta is more transmissible. But from a kind of epidemiological point of view, at this point, when everything is Delta, we want to be able to do subdivisions of Delta. And so right now there are about 120 subdivisions of Delta, and Thailand has two particular named lineages. We have AY30 and AY85, which are thought to be predominantly Thai lineages. And they are the majority of the Delta we're seeing in Thailand now, with occasional introductions from other countries. So I just wanted to highlight this because I should have put the link in the presentation, but this is on our website, and we try and keep this up to date as new data comes in, so that you can track all the cases that are being sequenced in Thailand. So I'll just go back to my presentation.
While you're doing that, Liz, can I ask you a quick question? Well, quick questions come in from the audience, and that's about selection of samples for sequencing. I mean, what kind of strategy do you? I mean, how do you do it? You know, how do you make your choices on which ones to sample?
Yeah, it's a good question, and it's partly aiming for a representative sample scheme, but it's also partly kind of pragmatic what samples are available. So we get a lot of samples from local hospitals. And so we do a lot of samples from Bangkok hospitals. And I didn't show it in this presentation, I ran out of space basically, but the sequencing from Thailand is very dominated by Bangkok. And that's partly because the outbreak, you know, there have been more cases in Bangkok than anywhere else, but it does make you worry that you are undersampling from other regions. And that turned out to be important because when we had an introduction of the beta variant, which is thought to have a reduced vaccine efficacy. And so it's important to be tracking that. It didn't arrive into Bangkok, it arrived into the south of Thailand. And there are occasional cases still from there. So it's kind of, we have what we'd like to do. I mean, in an ideal world, you probably set up a sampling, a sampling frame and do it, but there are issues with access to samples. There's issues, especially in remote locations, with managing to get the RNA to a central lab for sequencing on time if they don't have sequencing capacity there. And I think a lot of the sequencing is quite centralized in a few labs. So it is improving. We're getting a lot more sequences, particularly the Ministry of Public Health are doing a lot of sequencing from different provinces, but a lot of it is dominated by Bangkok and also Tak province, because we have a site in Tak province who send us samples.
Okay. Yeah, thanks for that. That's a good answer. So a sort of mixture of pragmatism and aspiration, if I can put it that way. Yeah. Okay, great.
Yeah,
Sorry to interrupt.
It's all right. I was just going to go back to my acknowledgement slide, which is to acknowledge particularly Phi, who keeps that next train build up to date and has done a lot of bioinformatics for us. A lot of collaborators who work with on the sequencing, a lot of the other labs who also submit their data that helps us put ours into context, and a lot of funders have helped out with this since March 2020. And I'll draw to a close.
Okay, excellent. Well, as I said, we had one question. I'll just ask the audience if there's any other questions, to put them in the question and answer box, and we'll try and deal with them either as we go or at the end of the session if time allows. So great, Liz, thank you very much for that. That was excellent. I mean, it's fascinating, absolutely fascinating, the way the virus develops and mutates, and actually amazing the way that we can track it to such a detailed degree. So excellent. Thank you so much for that. I'm going to pass on to Christopher in a moment, but I have a quick poll to launch on Christopher's behalf. So bear with me. I'll do that now. But thanks for that, Liz. Let's put this up. We're going to ask you a quick question about the molecular tools that you might use in your own laboratories. So here we go. So which of the following molecular tools do you use in your laboratories for the detection of pathogens in clinical samples? So traditional PCR, real-time PCR, Sanger sequencing, whole genome sequencing, metagenomic sequencing, or none, if you don't do that. So we'll wait for that. So Christopher, go on. What's your guess? What do you think is going to be the top answer?
My prediction would be real-time PCR. It's the sort of most common technique that's used around the world really these days. But we are seeing more and more sequencing beginning to appear in labs, and that's a good thing. And yeah, we also still have some traditional PCR happening where things can't be transferred to real time. So I'm hoping it'll be a bit of a mix.
Yeah. And you've been, while this is coming, you've been doing parallel work to what Liz has been doing in Thailand, in UK HSA, Porton Down. I mean, you've been doing sequencing there. Same kind of work, I guess. Same kind of thing.
Yeah, very similar. Whole genome sequencing, and like Liz said, the important thing here for everyone is that all the sequences that we do produce, whichever lab it is, wherever it is, all of those are uploaded onto the GISAID platform, because detecting these variants, you can't detect them unless you know what's circulating. And if we were to sequence things and not share them, then that's it, slowing that whole process down. So it's a massive team effort across the globe at the moment. And as Liz showed, over 5 million genomes have been uploaded. That's huge.
I mean, and the UK's sampling strategy, how does, I mean, the question I asked to Liz, I mean, the question was, how do you select your samples? What's kind of...
Yeah, I mean, the strategy sort of adapts to the situation. So depending on how people are being tested to start with. So for example, people coming in through airports, etc., would have to do testing, or any positives from those kinds of samples would probably make it into sequencing. So yeah, there's a wide range of strategies, but those strategies do change as the environment changes, really.
Sure. Well, anyway, here's the results of the polls, and you were spot on. 91% using real-time PCR, but a mixture of the others as well. So yeah, so that's good to know. Yeah, okay. Well, thanks very much for everybody for answering the question. I'll stop that and hand over to Christopher to fire up his presentation. And Christopher, you might as well say a little bit about yourself, because I've neglected to get a bio from you, but I know you're.
Let me just get
there's your title. Yeah.
Is that, have you got the full screen, or do you have?
Not yet. No, you need to click on the presentation button. I think it seems to be just. We've got the little view of the slides down the left hand side.
Oh, yeah. Okay, sorry. How about now?
Yeah, that's better. Yeah, perfect.
Yeah, so thank you for the invitation again to deliver today, and yeah, I'm looking forward to discussing and hopefully, if we don't have time for questions today, at least answering questions later if we run out of time. Yeah, my name is Christopher Logue. I work at the Novel and Dangerous Pathogens training team in the UK. Our real role is providing training for high containment laboratories, BSL-3 laboratories, working in virology, bacteriology, molecular biology, a whole range, biosafety, biosecurity, whole genome sequencing. So a whole range of techniques and laboratory levels. We provide training to scientists here and around the world, so that that work can be done safely, and that we're also maintaining a high standard when it comes to quality control of things like sequencing or PCR and things like that. So a mixed bag. But today, actually, a lot of the work I'll be talking about today is actually from my previous PhD student who just got her defended her thesis just last year about using metagenomic sequencing as a mechanism of identifying new diseases. So I'll talk a little bit about that. Okay. So, sorry, my very patchy computer. So yeah, with any kind of sequencing, we needed something to see. We need something to sequence, and then, unfortunately, there's no shortage of pathogens around the world at the moment, and re-emerging pathogens especially. But the WHO define emerging infectious diseases a disease as one that has either appeared and affected a population for the first time. So, for example, SARS-CoV-2, or has existed previously but is rapidly spreading. So a good example of that might be Zika. And that's either in terms of the number of people getting infected or to new geographical areas, or indeed both. So what we really see is that we're very much interconnected, but with an emergence of a disease, it starts in it has three real phases. It's a pre-emergence, where it might pass from one host or one reservoir to another, and then that becomes a localized emergence, where it might jump from, it might be a zoonotic disease where it jumps from an animal to human. And then finally, we have our pandemic emergence, where it's transmissible between humans. So we're very, very interconnected in terms of species, in that we are moving and encroaching more and more on animal species and their spaces and their environments, their habitats, where humans are becoming more in contact with pathogens carried by those animals. So this One Health approach that the WHO and a lot of we're taking is key for our future, really. But then, as you see on the right, we're so much more mobile now than we were 50 years ago. And on the right, that's just a map of the flight paths around the world. So just because something happens in a remote area does not mean by any means that within a few days it could be across the other side of the planet. So there are many global diseases. Just this word search, Brady sort of word bubble shows just a few of them. They're distributed around the world. This is based off search engine results at a various period of time. And they can really come out of nowhere. This is from February 2006. I did my PhD on a back in 2002, I think it was, on an unknown virus no one had ever even heard of, called chikungunya virus. And I was very proud of myself at the time, because I was the first person to sequence this virus that no one would ever study or look into other than me and a couple of other people. And then suddenly in 2005, 2006, this little virus would start to change the way we look at diseases. When I finished my PhD back then, I moved to CDC, and we just because this virus was emerging, we put out a paper at the time just to bring it up to share with others. And you'll notice from our very basic map here was that South America wasn't even on our map, because there were no cases in South America. A little can change a lot, and this is where sequencing again starts to introduces itself, and a single amino acid change in the virus of chikungunya suddenly allowed it to then start to replicate in other mosquitoes. Previously, it had only been really seen in Aedes aegypti. And within a short period of time, this virus was able to essentially move itself around the world, transmitting between individuals who were then returning to home countries and then getting bitten by mosquitoes, and this continual transmission. So from very few cases to suddenly being this little unknown virus suddenly became a huge problem, as many others have as well. So it just shows you that if we don't know the sequences of some of these pathogens, we're not going to detect the importance of these mutations. since then, there's been an awful lot. We've had Zika, we've had SARS, we had SARS back in 2002, and we've had several Ebola outbreaks and Zika and monkeypox most recently in Nigeria, all of which have kind of been eclipsed by COVID over the past 2 years. But a lot of these diseases are still ongoing, and we're still seeing them throughout COVID. They haven't gone away, and they won't go away. So with so many illnesses and so many potential positive agents, we really need to be able to diagnose well. And a lot, especially arboviruses, which is viruses spread by arthropods, mosquitoes, etc. A lot of the initial symptoms are the same. It's very difficult to differentiate between someone with dengue, someone with chikungunya, and someone with malaria at the early stages of the disease. So this is where we have to look at some of the diagnostic options. So option one, which is very important, is the clinical history of the patient. When did the symptoms happen? Anything that might have led to them? So that's still very important. We've seen an increase of rapid diagnostic tests. These, while an option, by no means are the most specific or the best. Serology, this is already looking at if someone's been infected. And then, as many of you showed in your diagnostic labs, 90% of you are using PCR, which is the gold standard. And we're introducing now targeted genome sequencing, which will allow us to know more. But this option 6, metagenomic sequencing, the one thing where it differs from the others, from the other molecular diagnostic techniques, is that it's unbiased. We're not looking for a specific pathogen, we're looking for any pathogen. And while it has its drawbacks and its complications, it is very much, it's a hugely useful tool in looking for things we might not otherwise look for. So I think that the key message here is that a combination of some of these is most important. So, for example, in the onset of a particular disease, we have the viremia. So in this case for malaria or chick, for example, we have the viremia, and only when there's enough virus in the blood can then our PCR pick up virus genomes, and then after our antibodies start to clear that virus, then we can start to detect higher levels of antibodies. So traditionally, this is sort of onset for an arthralgic disease. But now we're looking at genome sequencing, because we can start looking, as soon as we see any viremia, we can start looking for the genomes of the pathogens within that. So this talk's really based off, like I said, my PhD student Emma Wise, her research, which we had a case study. We had a 41-year-old male who had fever, headache, joint pain, and nausea. This was all in 2006 during an epidemic of Zika. So that wasn't hugely surprising, other than the individual tested negative for Zika and negative for chikungunya and dengue. Sorry, he couldn't remember being bitten by mosquito. That's not surprising. But he had stayed within this area in the northwest of the country in Esmeraldas. So we decided to look for other pathogens at what might be the cause. And when we were doing this, there were another 195 other isolates from similar cases that we also looked at. So we had our 196 blood samples, all of them with a variety of non-specific symptoms, as you would expect with any arthrovirus, arbovirus, and during an epidemic. But in most of the patients' cases, there wasn't an actual clear diagnosis. So the clinical presentation, I said, it was fever, headache, joint pain, muscle pain, all of which could be associated with a range of different pathogens. So which ones do you look for? Can you look for them all? Do you have assays to look for them all? And this differential diagnosis is helping us rule out which pathogens it isn't, which is equally as important as detecting which pathogen it might be. But yeah, let's look at fever. Right, well, they all have fever. They all cause headaches. What about joint pain? Yep, all 4 of them cause joint pain, so we're not narrowing it down too much. Nausea, okay, we've got some vomiting, but that's for malaria and dengue, but that might, that's not necessarily a truly identifying factor by itself. So this is really where we get a picture of what the symptoms are, but we really need to have that level of screening to identify what it might be. So that's where the PCR came into it. We did a series of multiplex PCRs where we could look. We looked for the sort of usual suspects, the pathogens we thought were most likely to be circulating in Ecuador at the time. And of the 196 that we screened, we got 49 possible positives across 4 pathogens, so Zika, unsurprising, dengue, chikungunya, and lepto. The reason that we might have picked up Zika in cases where they weren't picked up at the hospital is just to the level of sensitivity to our assay was somewhat better than the ones that were being used in the hospital at the time across the board. Okay, so that was great. We were able to sort of say, okay, well, now we know some of the potential causative agents, even though some of them, as you can see from the graph, were quite late. CT values were quite late in infection, in the real-time amplification. So they were very, very weak to start with. But what strains were they? Is it a new strain of Zika? Is it a new strain of dengue? What was it? And what about the 147 samples that we didn't detect anything for? And that was the big question we were most interested in. And our case study, our individual patient fell under that. So with metagenomic sequencing, it's unique in that it's unbiased, as I said earlier. It sequences everything in a sample. So whatever is put in, it will sequence that nucleic acid. So this is great because it would capture everything, but also it's quite difficult because if you had any contamination, it will pick up the contamination. If you have any commensal bacteria, it will pick up that. So you really have to know what to rule out before you do a metagenomics sequencing analysis. But in a nutshell, the process works where you put all the genomes in there. The genome's fragmented into many, many small fragments, which are then barcoded and libraries are prepped, just like Liz described earlier. All of the fragments are amplified, and then through clever software, we remove the human reads, for example, because the majority of the DNA in a sample or nucleic acid in a sample will be from the patient themselves. So we remove that from the data set. We remove anything we're not interested in looking for, like commensal bacteria that wouldn't normally lead to a patient becoming ill. So we can start to, we bring everything in, and then we start to separate things out and see what's left behind. So for this work, we were actually, we only looked at 18 patient samples out of the 140 as a proof of principle. We selected some that were positive for dengue and some were positive for chick, some were positive for both. But the majority we looked at were negative for everything that we'd looked for, because that's what our question was. If it's not dengue, it's not chikungunya, it's not Zika, it's not lepto, what is causing this illness? So the steps for DNA prep, in this case, because we weren't interested in any pathogens or any viruses that were DNA viruses, we could get rid of, and we weren't looking for bacteria, we could look, we were able to degrade all the DNA in the sample. So that way we were only looking at RNA, which helped us narrow it down in this initial pilot. Once all of those sequences have been created, then obviously we have the analysis. There's several tools. I've listed them here, and these are very, very ingenious and well-written algorithms that allow us to go in and classify particular sequences into groups, and from that, then we can align these with all of the reference genomes that are available to us. So it's a series of algorithms that help us narrow down our search and allow our search to be faster and more efficient. So from the metagenomic sequencing, our individual was negative for everything, but had some very faint PCR signals for dengue and chick, but these didn't actually appear. We didn't get deep enough reads for either of those in our sequence. But what we did find was another RNA genome that we got quite a lot of sequence for, and this was a virus that previously hadn't been shown to circulate in Ecuador, called Oropuche virus. And we got some coverage over that viral genome, which when we looked and learned more about the virus and the diseases it caused, the symptoms it caused, made a lot of sense. So Oropuche is it's an orthobueniovirus. It was found in the neighboring countries to Ecuador. It's transmitted by a midge, but there was some speculation that it might also be transmitted by mosquitoes. It has 3 segments in its genome, and although there's been no evidence of it in the country, in Ecuador, there in earlier studies in the 50s and 60s, there were some potential cross-reactive antibodies potentially against it, and it can transmit in urban and forest cycles. So the workflow really that we had was we screened the 196 patients using the PCR panel that didn't include Oropuche. Through the metagenomic of a subset of those, we were able to identify Oropuche, and then what we did was we then went back and we actually looked and tried to test that sample using the published PCR assay, but it wasn't detected. So that was an initial fear and surprise. In order to be able to look in more depth, and also it's the sort of gold standard for any virology, that we tried to isolate the virus in vitro. So we grew up the virus, isolated it. Once we had that, we then had enough material to do whole genome sequencing, following the same process as Liz mentioned. And once we had the full genome sequence of this particular virus, we were then able to compare that sequence and compare the primers that were already out there for the published PCRs against our sequences. Once we had done that, we were able to then look at how we could optimize and tweak those PCR conditions. And then we went back and tested all of the 196 patients using the new PCR assay. And that showed us 5 additional cases which were also missed. So like I said, we tried to do this PCR using published primers and published PCR, and yeah, this was a point in time where the assay wasn't picking up our sample. I think there's a poll question. We might just leave that to the end, okay? I think it might be easier, if that's all right, John.
Yeah, that's fine. I have a quick question coming in. You might be able to cover it quickly anyway, Christopher. It says for a country which has no record of of circulating genotype for Japanese encephalitis virus, what is the best method for genome surveillance? And I guess this metagenomic technique might work. Does that? How would you see that?
Well, I mean, if I think already PCR would be quite effective. You would keep it as part of a panel, a differential diagnostic panel that you would use. You could continue using PCRs. If you were getting negative for that and negative for other things, you might then come down this route. Yeah. So
you've got a known. This is a known virus now, isn't it? So presumably, it's a PCR would be set up to pick that up. Yeah.
And once you've identified something, sequencing it is very straightforward, and then you can design a new PCR or better PCR, which is exactly what we did here. We designed a new PCR, and you can see on the graph on the left here, the original PCR in green picked up the virus in the Ecuador strain, but only in very high concentrations, which is why we missed it. Whereas the two primers we designed 2 separate ones, and you'll see that both of them on the left pick up the Ecuador strain very, very, very well at very low levels, but when it comes to the prototype strain, other strains of Oropuchi virus, it wasn't great at picking up other strains. So the one of our primers didn't pick up a lot of the other strains, the prototype strains. So, but one of them did. So that's the one that we used then to retest samples. It picked up the Ecuador strains well and prototype strains, and we could align that to see what other strains it do. So that identified six patients. Obviously, every assay that you develop, you have to test against a whole range of other pathogens just to make sure that it doesn't pick those up, that it's specific to the virus or pathogen you're looking for, and not picking up other pathogens. So we isolated it. We isolated the virus. We were then able to sequence it, and as soon as we possibly could, we made that genome sequence available freely on GenBank, and we sequenced it using the normal whole genome sequencing. Once we had done this and we compared it to all the other existing strains that we had available at the time, it was most closely related to a strain that was circulating in Peru 8 years earlier, which is a neighboring country. So again, in order to alert neighboring countries and other institutions within Ecuador, this was also published quite rapidly to ensure that people were now looking out for this as we went into a new season of infections. We were able to isolate virus for all of the different patient samples by growing it up in cells. And then once we had done that, it allowed us then to use whole genome sequencing to to fully sequence in good depth each of those patient strains. And then we were able to release those so that individuals could look to see what changes there were to the actual genome, if there were variants, what sort of variation there was between strains. During this time, other groups, colleagues in Oxford, were also looking at sequencing a lot of different strains of Oropuche, which is great. So we worked closely with them and included our strain, as you can see up at the very top right of the screen, with their considerably larger phylogenetic analysis, and it was identified that they formed a single lineage that was most closely related to the Peruvian strain of 2008. So the summary really here was, it's all about an integrated approach. We've got the clinical signs, the history from the patient. You know what you expect to see, and that's really what PCRs generally are all about. It's you're looking for something you're expecting to see. And if that comes back negative or even positive, if that question hasn't satisfactorily been answered, then we could look at metagenomic sequencing, where we're not just looking for one pathogen, we're looking for any pathogen. Through the analysis, then we can identify if anything is there in the background that we weren't expecting, and identifying that through isolation provides enough nucleic acid for us to do full genome sequencing. And then comparing the existing molecular assay tools, so all the primers and probes on the market for that pathogen to the new strain, we can see if that those primers and probes would indeed pick up this strain. And we can improve that. So finally, the other thing then is really what we also wanted to check was we were able to identify this in humans, but the actual Culocoides midge that is known to transmit this virus just isn't really found in Ecuador, in that region of Ecuador at all. So the question was, well, how was it transmitted? So we know what species of mosquitoes are in that area of Ecuador. So we looking at cell lines, we were able to see if the virus was actually able to grow in those, and sure enough, it was both. Yeah. So that's something again that will add to public health decisions. So that's it really. Thank you again. As I said, this was happy to say now Dr. Emma Wise, who was a great student, and the sequencing team at University of Plymouth and USFQ in Ecuador, and the UK Health Security Agency.
Great. Christopher, I've got a couple of questions coming in, so thanks very much for that. I think you might actually have covered one already, but let's. So I've got a question that came in from Grace Gan in Singapore. It says, "Hi Christopher, what kind of cell line did you use to grow the various viruses? And also, if virus isolation is not possible, how do you ensure sufficient material to go into WGS?"
Yeah, both great. Well, the second question is really, really important. The first, yeah, we just grew these in Vero cells. We tried it in Veros, we tried it in BHK21s. So, and we found it grew best in Veros and mammalian cell lines. It also grew in several 80s mosquito lines, but there were several mosquito lines that didn't grow in. So part of that was really actually test and try and see what does grow, see what doesn't. As for the other question, yeah, that comes down to how you can prepare your sample for nucleic acid extraction, and how best to amplify up as much of the genome as you can. That's there's a long answer for that, which would require probably a blackboard and a lot of coffee, but I've left my details here. I'll happily have that discussion with you offline, but it's a really important question that you're asking. It's a very good one, because it is tricky sequencing directly from a sample.
Thanks, Christopher. Well, yeah, Grace, if you want to get in touch with Christopher direct, please do that, and I'm sure he'll answer the question in a bit more detail. The other question that came in, I think it's you sort of covered it, and it says, regarding those presented arboviruses, did you try doing metagenomic sequencing using mosquitoes or vector samples? And I think you kind of covered that.
It's something we want to do. It's something we will do. At the time, it was already the time had passed. Those mosquitoes these were samples from 2016, and we started sampling them in sort of 2017. So yeah, it's something we're going to go back and do. We're working with the university in Quito to go back and do that. But this was just a sort of pilot. But yeah, very important point.
So that would involve actually capturing the live mosquitoes and taking a sample from them. Is that how you?
Yeah.
Okay.
CO2 traps and then pooling them and then just extracting nucleic acid.
Okay. Great. Well, thank you very much. Thank you for the questions. As I say, keep them coming in, and Christopher details are there. Also, a reminder that on one of our previous webinars, we had an e-learning package for laboratory technicians, which is live. If you want to contact Christopher for that, that's also still possible. So, thanks very much. Okay, so. I think, Christopher, you close your screen share, and I'll hand over to Joris. So welcome to Joris from RIVM, and you're going to have a look at wastewater sampling, detection, and some sequencing in that, Joris.
Yes, thank you very much, John. Also, thank you to all the participants for joining this webinar of the P81 project in Southeast Asia. So I will be presenting about wastewater and using it as an early outbreak detection and surveillance mechanism. So the topics that I will cover in this presentation is first, what is wastewater surveillance? What are the benefits of it? But also, what are the challenges of wastewater surveillance? And why do you still need human surveillance? Then some examples of pathogen detection using wastewater surveillance, and the integration of this surveillance technique in pandemic preparedness. And finally, I want to advocate harmonizing wastewater surveillance for multiple pathogens. So wastewater surveillance is the process of monitoring wastewater for biological, chemical, or other contaminants. And examples of biological contaminants are different viruses and the bacteria, such as poliovirus, SARS-CoV-2, but also bacteria with antimicrobial resistance. It's also used to monitor drug secretion. So this can be licensed drugs or unlicensed drugs. One of the applications is to see if there's a black market for certain drugs and see if the amount of medicine, for example, that is predicted based on purchases by hospitals and pharmacies, if that is similar to the medicine concentration found in sewage. You can also measure consumption, such as coffee, or more likely caffeine, which is secreted in urine. But also risk factors for a disease, and this is quite a novel area where certain risk factors for cancer, for example, or obesity are monitored in wastewater. And as you can see, that if it's secreted in stool or urine, then it is likely that it can be detected in wastewater surveillance. And that is particularly interesting for pathogens, novel pathogens, that if you can detect it in urine or stool, you can likely also detect it in wastewater surveillance. So wastewater, there are several contributions of different sources to wastewater. So of course, humans making use of the toilet, but also animals can contribute to wastewater. And for example, the sewage drain of slaughterhouses that can also be contaminated with animals. Industry, so large industry plants also use water for their processes, and they can discharge this wastewater in the sewage. And weather, so rain can be definitely a factor, and that can dilute your wastewater, for example, from households. And therefore, it's also very important to know from your sampling area if it's mainly humans via households that contribute to the sewage you're measuring, or that also large industry is connected to the same sewage, and also that you monitor the weather and take into account rain events. And one of the ways that is being done is by measuring the amount of water that goes through the sewage. Well, then there are different places where you can sample water. So that can be at the wastewater treatment plant. You can see that here on the right. And at these wastewater treatment plants, we have special devices that collect wastewater for 24 hours and combine that into one bottle of half a liter to 1 liter. You don't need to have a sewage system to have a wastewater surveillance. You can also use open drains in certain neighborhoods, or you can use even contaminated surface water. And South Africa has some beautiful reports on how they detected SARS-CoV-2 in contaminated surface water. You can also sample at a much smaller scale. So at hospitals, for example, slaughterhouses, schools, or nursing homes. And also a novel one is in transport. So there was a publication last year where they sequenced SARS-CoV-2, or sorry, they performed a PCR on the toilet in aircraft carriers, but also cruise ships. And they mainly did that to determine which are high-risk countries and where do introductions of SARS-CoV-2 in the country come from. So what you can see in all these cases is that these are collection points of wastewater, and these are usually good places to sample for wastewater surveillance. Well, there are some important benefits of wastewater surveillance compared to other types of surveillance for infectious diseases. So first of all is that the large amount of individuals is captured in one sample. So for a wastewater treatment plant, usually 1,000 to 100,000 individuals are connected to that wastewater treatment plant. And it means that a significant proportion of the population can be sampled or can be surveyed with only a few samples. So for example, for the Netherlands, we have 17 million inhabitants and almost everyone is connected to the sewage system. And we have 300 different wastewater treatment plants. So by doing 300 PCRs a day, we can perform surveillance on the entire population of the Netherlands. It also includes pre-symptomatic and asymptomatic patients. So that's a difference with human surveillance is that most often in hospitals, people only go there when they have symptoms. While with wastewater surveillance, you also include those persons. And also enabling screening of rare patients. And this is mainly if you do large population studies, but but only a very small percentage of the population has the pathogen. That means that you have a very large study population of maybe 10,000 or 100,000 individuals to just get enough information on those rare pathogens. And there is also where wastewater surveillance can be of use. Because you only need to test a few samples to cover a large population, it's also cost effective. So doing 300 PCR assays, for example, a day for the Netherlands, it's much more cost effective than trying to do 17 million. You also circumvent sampling of individuals. So it's non-invasive. For SARS-CoV-2, it's a nose swab. Which is an invasive technique, but can also imagine that you have to draw blood or those kinds of procedures. And because almost everyone makes use of the toilet, there's also no selection bias in your sampling technique because you almost cover the entire population except for people who are wearing diapers. There are also certain challenges of wastewater, and the biggest challenge in wastewater is that you measure a concentration in wastewater. For example, in virus particles per 100,000 inhabitants. That's currently how we depict the numbers in the Netherlands. But how do you then translate that in how many people of those 100,000 are affected? And therefore, you would need to have really detailed knowledge on the excretion of the pathogen in symptomatic, asymptomatic, and pre-symptomatic carriers. Well, you need to know the size of the underlying population and the movement during the day. They can be connected to one wastewater treatment plant with their house, but when they go to work, they might go to a different area with a different wastewater treatment plant. You need to know the design of the sewer network. So which household and which building is connected to which wastewater treatment plant, but also the fate of the agent. And what we mean with that is that some viruses are very stable, such as poliovirus, but some viruses are unstable, such as SARS-CoV-2, and likely the concentration that is discharged into the sewage by the individual, that SARS-CoV-2 will deteriorate over time before you sample it. Then, as I mentioned, the influence of rain events on concentration, and particularly for bacteria, is also the mobilization of a biofilm and sediment. And then detailed data on the signal loss during analytical procedures. So you need to have extraction controls, concentration controls, etc. Then the examples of wastewater surveillance. So wastewater surveillance has been used the most for polio. It is strongly embedded in the polio eradication program. Polio is currently only endemic in Pakistan and Afghanistan, and outbreaks are detected using wastewater surveillance, and they are then used to start vaccination campaigns. Well, in the Netherlands, we sometimes have introduction of poliovirus. There is a certain area in the Netherlands with very low vaccine coverage of polio vaccine due to religious reasons in that region. And in schools, we sample wastewater, and when we detect it, we start a vaccination campaign in those schools. And the reason why polio surveillance is also so successful in wastewater is because we know the sensitivity, and we know that the sensitivity is almost higher than in individuals. Because with 100 infected asymptomatic individuals per 10,000, you can detect it in sewage. And the problem with polio is that a lot of people are asymptomatic. So it will take some time before the first individual will seek medical attention, and then probably it's already much larger. And what is unique in polio wastewater surveillance is that it is directly linked to public health interventions. So detection in the wastewater surveillance leads to a vaccination campaign, and that is quite unique. Then for SARS-CoV-2. So since the pandemic, groups, countries, organizations have started SARS-CoV-2 wastewater surveillance around the globe on all different scales. Our institute, RIVM, performs surveillance on all wastewater treatment plants in the Netherlands. So these are 300 wastewater treatment plants. And these results are presented on the Corona dashboard. So this is a dashboard by the Dutch government, and it shows the number of cases, number of hospital admissions, number of admissions at the ICU unit. And here on the right, you can see the graph of the concentration of viral particles in the sewage per 100,000 inhabitants. And this is from September 2020 till now, and you can see that we are currently in a huge wave. We have currently, I think, 23,000 positive cases a day, and around almost 25% of people tested is positive. So it's a huge wave at this moment. In addition, we've done pilot studies in schools and nursing homes. Well, what we detected in schools is that wastewater surveillance did not perform better than human surveillance. So meaning students with symptoms would go to a testing clinic and get tested. While in nursing homes, we were successful. So we have a very nice case study where in a nursing home, we sampled the wastewater. No cases were known in the nursing home. And then when the wastewater, we tested positive, we informed the health services, and they tested everyone in that nursing home, and they detected an outbreak of between 10 and 15 cases in that nursing home. And they isolated everyone, and in that way, wastewater surveillance preceded human surveillance. And we think we weren't successful in schools because, well, you need to make use of the toilet, and otherwise you cannot detect it. And we think that that many students do not make use of the toilet during the day. Well, what we've also seen with our studies in the Netherlands is that the concentration of the virus in wastewater correlates very well with human surveillance, and possibly even precedes with 1 week the human surveillance. So that's very positive. And we are now currently modeling that to see if we can really use SARS-CoV-2 as an early warning for the human cases. Then antimicrobial resistance. Also wastewater treatment, wastewater surveillance is performed on antimicrobial resistance. And that is to determine if discharged by wastewater treatment plants contribute to the presence of AMR in surface water. And also to investigate if sewage surveillance is useful in addition to human surveillance. And the way that's being done is with CPE producing enterobacteria. So these are critically important according to the WHO. And community carriage is difficult to assess because of asymptomatic individuals. And we did a study of several years in the Netherlands, and what we see that over time, the level of CPE producing enterobacteria increased. We also saw that there was a correlation with hospitals in the surface area. So if a hospital discharged in a wastewater treatment plant, those levels were usually higher than when there was a wastewater treatment plant that was not connected to a hospital. But still, we also sampled the hospital to see how much they then contribute. And these hospitals explain only 25% of the load. So most of these CPE producing enterobacteria are in the general population and do not come from the hospitals. And also interesting, we see that the loads were higher in urbanized areas compared to areas with less urbanization. So then we have a few other examples. So there was a gastroenteritis outbreak in nursing homes. And we discovered the identical virus in the sewage as well as in the patients. Same was for hepatitis IE virus, also detected in surface waters and in human cases. Well, we also performed a study on the norovirus outbreak associated with a recreational lake in the Netherlands. And what we discovered is that this was not influenced by external human fecal sources. That meant that there was no sewage discharging or leaking into this recreational lake. And also another bacteria, Francisella tularensis, could be environmentally tracked by genotyping in water samples. And we showed that it was also successful when there are very low levels associated with human cases. So wastewater surveillance can be quite sensitive. So now in wastewater surveillance in pandemic preparedness and response. So you can perform targeted surveillance. What we are currently doing in the Netherlands is that we perform whole genome sequencing on around 70 locations to detect the introduction of new variants and also the spread of variants. And we're screening in wastewater also for known pathogens that are not endemic. So for example, for polio, this is not endemic in the Netherlands, but it is sometimes introduced by travelers or by immigrants, and we detect this introduction in the wastewater, and this is targeted using real-time PCR. But of course, what you would like to do for pandemics in the future with unknown pathogens is using untargeted surveillance, as my colleague Christopher Logue explained. And in wastewater, that's a very challenging approach because you have no clinical symptoms that you can link. It's completely unbiased. But it is possible, and there's a very nice study on the global monitoring of antimicrobial resistance based on metagenomics analysis in urban sewage. And what's very important is that if you perform this unbiased surveillance in wastewater, is that you also integrate it with other surveillance programs. And a recent study that explains it nicely is from the group of Marion Koopmans in the Netherlands. Is on a One Health perspective on preparedness. So that's also surveillance on slaughterhouse waste, but also surveillance on sewage of live animal markets, for example. but also connected with this human surveillance. I mean, also this pandemic of COVID-19 was detected because of humans seeking medical attention. And if you have a detection of a novel pathogens, then whole genome sequencing is very important, as Christopher also explained, so that you can design new primers to detect it using RT-PCR. So just a couple more slides. This is on the early response timeline in the Netherlands. So on the 11th of January in 2020, the sequence of COVID-19 was first known. And on the 4th of February, the primer assay was published by the American CDC. And we used this primer assay on wastewater. Then on the 27th of February, the first case of COVID-19 in the Netherlands was detected. But there was already, we reached our peak in March, and probably in February, there were already thousands and thousands of patients. Because also at that time, I think we only could test a couple hundred people per day. So we were completely overwhelmed at that moment. And a day later, the first 2 wastewater treatment plants were sampled. This was extended from April till June to 30 locations. And after June, we started sampling all 300 locations in the Netherlands. And I think what's a very important question, if you look at this timeline of the early response, is why only 2 locations? At the end of February, and also why did it take a month before taking the first samples? And the reason for that is that we perform a lot of wastewater surveillance in the Netherlands, but for AMR this was a project lasting for several years and ending in 2019. So there was no ongoing wastewater surveillance for AMR, so we could not use samples collected for AMR to test for SARS-CoV-2. And therefore, it's very important that you have continuous sampling. And the second reason is that we perform continuous sampling in schools in that region with very low vaccine coverage for poliovirus, but that is only on schools. And on schools, we did not think it was a suitable location to test for SARS-CoV-2 in the beginning. So the second is that you should select locations that are useful for multiple pathogens. And then the third one is store samples and create a biobanking so that you can do retrospective analysis. So in the Netherlands, when that first case was detected, if we had wastewater from different cities and we would look a couple weeks back, we would probably see that it was already spread between multiple cities in the Netherlands. And a nice example of how you can do this, uses retrospective testing, is performed by Pakistan, who use samples of the polio surveillance program for SARS-CoV-2 detection. Well, having continuous sampling can be quite challenging because costs are involved, and that's also the reason why our AMR surveillance is project-based. For example, every time there's a new research question, there's a new project. The moment that a question is answered, the sampling stops and it starts again. So how can you do continuous sampling and also make it cost effective and be prepared for future pathogens? And that's why we are currently writing a guidance document on a harmonized approach to wastewater surveillance. So instead having separate surveillance programs on wastewater, where each takes care of their own governance, on their own data interpretation and reporting, trying to connect with health services for vaccination campaigns, for example, is try to combine that all in a harmonized approach where you have SARS-CoV-2 monitoring, polio monitoring, and AMR, and also future unknown pathogens that is all integrated into one program that's well connected with health services for potential health interventions. Because the reason why it took a month before we had the first samples is because Well, we first had to contact the wastewater treatment plants. We had to ask their permission. We had to contact with the Ministry of Health in the Netherlands. So that all takes time, and having a continuous system will make sure that you can have this rapid response. So with that, I would like to end, and I would like to thank the Dutch water boards who provide us with all the samples. Also the head of our department, Anne-Marie de Rode-Hasman. And I've presented the research of the other people in this list. And if you have any questions, please contact me on my email address shown here at the bottom of the slide. So thank you very much.
Joris, thanks very much. In fact, we do have a couple of questions which maybe you can answer now. So I've got one from Hazik Mumin in Brunei, and asks, are there any guidelines on processing wastewater after collection but before molecular work? Is there some guidance available, or maybe regulations almost?
Yeah, there is a guidance document for polio wastewater surveillance from the WHO. If you Google WHO guidance document, polio environmental surveillance, I think it's the first hit. And that is a complete guidance document from taking samples to storage and analysis. So you can find guidance in there, and also in our new guidance document that we are writing on a harmonized approach, we will also cover that. Okay.
Great, thanks, Joris. So, Hazik, I hope that answers the question, but the WHO site seems to be the best point of entry for that. Second question is from Yang Lian Hing in Malaysia, and it says, the question is, the virus could be detected in wastewater, but would not be infectious. So are you just checking for the presence of the virus rather than an infectious dose?
Yeah. Yeah, for SARS-CoV-2, so poliovirus is infective when it still is in wastewater, but SARS-CoV-2 is not infectious when it is in wastewater. And what we want to do with SARS-CoV-2 is really monitoring trends. So is there an increase in SARS-CoV-2, or when the government takes new measures, such as a lockdown or restrictions, or then can we see a decrease? So it's really to monitor the pandemic in the population.
Okay, great. Thanks, Joris. And Yang, I hope that answers your question. I think we're going to have to close. I think everybody is either feeling hungry or has gone to the toilet. I don't know whether that's a result of your presentation, Joris, but maybe if you just close your screen now, and I'll say farewell to everyone. Just a few last words. Massive thank you to all our speakers. Thank you so much, Liz, Christopher, Joris, Alex, for being having been grilled by me at one point. Sabai, thank you for helping me with some of the questions. That was excellent. I hope you all enjoyed it. The recording of the webinar will be put on the Project 81 website shortly. The other previous webinars are on there, which you can access them if you want to look at. Christopher's kindly offered to put a PDF version of his presentation on the website. I'll check with the others if they can do the same, if they're happy to share that. It's contained in the recording anyway. So thanks everybody for staying around. We've got another webinar coming up in December. I'll just give you the date now, and that is going to be the 17th of December, and we're looking at legislation. So completely different topic. We're looking at how the lawmakers have dealt with the pandemic and the types of techniques that have been used. So we've conducted a survey. So slightly different subject matter, and I suspect a slightly different audience. But on that note, look forward to seeing you again. There's a feedback questionnaire which will come out automatically at the end of the session. Please take a few moments to fill that in. It helps us immensely. And if you do, thank you so much in advance. So great to see you all. I'm going to close now. So on behalf of the team and all our panelists, thank you for attending, and see you next time.